Here with Gergo Orban, and we are going to talk about biological vision, which is actually a lecture that I'm super interested in. Gergo, on to you.
Okay, lovely. Thank you again for introducing me, and I apologize for these technical issues. So today I will speak about neuroscience, which is obviously a huge field, and I will just take one specific aspect on how machine learning ideas can be used in neuroscience, and how neuroscience will then be able to inspire research in machine learning. So my research interests lie in the intersection between neuroscience, cognitive science, and machine learning. Just to give you a slight background for this, traditionally cognitive science is considered to be the discipline to study behavior, mostly concerning human behavior, and neuroscience is concerned with any sort of neurophysiological signals that one can obtain from the brain. But obviously these two things interact a lot, and when you take into the picture of machine learning, then you are there, in my word, in this tiny little intersection between the three different disciplines. So instead of trying to cover all the possible links between neuroscience and machine learning, I will take one specific point of view, and I will try to navigate you through ideas how machine learning can be directly used in neuroscience research. To highlight some aspects of the problems that we are faced with, we can just look at this bunch of pictures, which are the original drawings of naive human subjects who were asked to draw bicycles, which, as familiar they might seem, are as variant across people how they actually draw these bicycles. And if we just consider this problem as a huge problem, whereby you learn about bicycles, then you try to reproduce these bicycles based on your memories, then one is tempted to think of this problem as something as a computationally challenging problem, and then we might want to access this problem by trying to building up computational theories, like whether the sort of errors that humans make when they draw bicycles are connected to lossy compression using the semantic word model. Well, this sounds like a long shot, but the main point here is that the sort of patterns of errors humans make might highlight these sort of computational principles, one of which would be performing lossy compression in the brain. Just to show sort of an example from the opposite end of the spectrum, one might have these experiences when episodes of their life come out very vividly, and one can describe a specific scene from their own life with some staggering amount of detail, which might sound like a promising thing to do from your memory, because, well, a faithful memory is the best memory. But on the other hand, if you consider this from a computational point of view, storing data points like this in great detail is not really a great strategy, right? If you want to generalize to some extent, this is not really a good idea to store highly detailed memories in such a detail. And again, one might allude to theories like continual learning or Bayesian structural learning to try to explain why people tend to maintain these memories, and what sort of dynamics one expects from these memories once additional experience is accumulated. From the domain of vision, I can illustrate our problem in a way like this. So if you look at this ceiling of a church, you see pretty much a two-dimensional image, and from this image you infer tons of different things. Your brain works hard to interpret everything on the scene. And again, one might come up with theories whether what we are doing is an approximately probabilistic inference in a hierarchical model. So I will then take this example vision, and we'll try to motivate alternative computational frameworks to address the problem of human vision. And we want to see how much we can learn from these computational frameworks about the computations that are actually taking place in the brain. So here's a sort of a cookbook for how neuroscience theory can work in the context of machine learning. So one might want to identify a problem that is ecologically relevant, then identify a computational formalization of this problem, and when we identify this computational formalization, then the complexity of the environment and the complexity of required behavior is required to be taken into account. Then from this point, we might want to derive an optimal solution for this problem, and then we argue that since the brain is an adaptive system and it had a couple of million years to do the best it can, so we assume first that there is an optimal adaptation that has been taken place in the human brain. And then with this optimal solution, we might want to formulate predictions for instance, for neural response statistics. Then we can test these predictions on recordings, and whether these predictions, so the level of depth these predictions hold, we can then go back to the formulation of the problem, to the formulation of the computational framework, and try to understand what sort of computational constraints there might be that affect the sort of representation that neurons build up. So just to make this problem more concrete, let's consider this issue. So imagine a natural image like this photo of a zebra. Then we can come up with two different frameworks. One is, let's call it deep discriminative models, whereby the idea is this: we have a bunch of pixels. Then these pixels are used to be able to make decisions about whether the animal that is presented on this image was a zebra or a horse. And to achieve this, we will use a bunch of hierarchically organized layers, and in between these layers, we will have a bunch of nonlinear transformation that enables us to make this decision. On the other hand, in the generative models, what we assume is that what we need to actually address, the problem that we need to address, is that we have observations like pixel activations, and we want to find the sort of causes that underlie the patterns of activations that we see in the pixels. So we want to find those latent variables that we do not directly observe, since we only observe pixels, and we want to understand how these latent variables contribute to the emergence of the specific image that we observe. These latent features are again organized into hierarchical layers. Here again, we expect some sort of gradually increasing complexity of these latent features. But at this point, we want to understand, so how can one differentiate between these two frameworks, apart from having arrows going in one direction or the other direction? So let me try to walk through this quickly. So one can identify the competition goal in the two cases. In one case, for deep discriminative model, the goal is classification. In the case of the deep generative model, it's about learning the data statistics. Then we consider what sort of architecture we are using to perform achieve this computation goal. Then we end up with a hierarchical feed-forward architecture in the deep discriminative model. And in the case of the hierarchical, in the case of the deep generative model, we have hierarchical model again, but because of the sort of arrows that go in the direction from deeper latent variables to shallower latent features, there is inherent top-down connections implemented in this framework too. The specific features or specific characteristics of these top-down connections will be highlighted later. The learning in the two cases is in one case is task specific. We can call it supervised learning in the case of the discriminative models, or task general, a non-supervised learning in the case of the generative model. And finally, some subtle little detail which we will go to in a minute. It's whether the representation that we obtain is probabilistic or not. In unsupervised learning, you might have encountered this issue whether one has a probabilistic representation or not. We will highlight this distinction later. So first, we will focus on discriminative models. How this framework can be used to understand anything about how the visual cortex works. So first of all, we use natural images to train these models. So the idea behind this is that it is the natural image statistics that constrain most of the parameters of the model. And since we can assume that the same statistics is the statistics that the nervous system has been adapted to is the same as the one we are training these models on, this way. This match between these natural statistics will sort of propose a constraint on both our model and our biological system. So for this, pretty much the standard tool that deep learning networks use is the ImageNet database, which are image patches with natural objects on them. The second component is sort of task that we are doing. It is classification. So on one hand, having a task-driven model is unnatural because what we are saying is that the model is actually adapted to one specific task. On the other hand, one can motivate this choice by sort of constituting a constrained Turing test for our model. A Turing test is a test devised in the 19th century to test sort of the level of intelligence of an artificial agent. And the Turing test has a binary outcome whether the artificial agent behaves as if it was a human or not. In this case, since we have a particular task, classification of images, one can come up with some sort of a human benchmark performance, which is actually coming from Andrej Karpathy, who was our hero to actually perform this classification task on thousands of ImageNet images. And this way, he set sort of a human benchmark on what reliability of classification we could obtain from humans. And as you see from these performance measures for different computer vision models from different years of the ImageNet classification contest, one can see that as time progressed, by the year of 2015, we got to the level with our artificial agents, with these computer vision tools, where we are pretty much at the level of human performance. So this way, at least in this very constrained task, sort of the machine matches human performance. So one can say that it passes the Turing test. The third component is hierarchical organization of the model. So because the structure of the visual cortex is hierarchical in the sense that the stimulus that we obtain through the retina goes to a sort of a relay station, the thalamus, the lateral genetic nucleus, and from there it arrives to the cortex. And even at the cortex, there are different processing stages, like the primary visual cortex, or V1, the secondary visual cortex, and we could go on. But luckily enough, things get more exciting by the time we got high in the level of hierarchy. We don't have the infinity. Rather, we have something called the inferior temporal cortex, where where neurons are sensitive to really high level features like objects and faces. So again, this way we might want to have a framework in which we have a similarly hierarchical organization as the human brain. So this is fulfilled in these deep learning models, whereby we have a large stack of layers through which these natural images are fed through, and then a challenge could be to identify the sort of layers in this artificial agent, the deep learning model, which correspond to different levels of activations in different neurons in different areas of the visual cortical hierarchy. So what you see here is RGC is in the brain, which corresponds to retina. LGN corresponds to the thalamus, and from these colorful layers of the schematics of the visual cortex, V1, V2, V4 are cortical areas. The last three are different parts of the inferior temporal cortex. And so we want to establish a correspondence between the model and the neural activities. So the thing that we allow is a mapping between neural activations Y to the deep learning model activations Y to the neural activations R. So we have a matrix A, which is just a linear mapping from these two. So we can linearly mix the artificial neurons activities to obtain a best fit for our real neural activities. Okay, so pretty much the matrix A, the linear matrix A, is the thing that we can fit. Now we can do this fitting, and then we might want to test how our model performs when new images are shown to an animal. So here we are considering a monkey, and from the inferior temporal cortex, or V4 of the monkey brain, activities of neurons are recorded, and we want to first we identify these neurons with a mixture of artificial neural activations through the matrix A, and then with this we want to predict neural activations in new images. So the question is then, if the performance of this model is comparable to the human performance, whether the building blocks of the model correspond to the building blocks of the models of the visual system. Sorry for the repetition here. So prediction. So we train our sort of matrix A on a bunch of images, like a couple of thousands of images, and then predict neural responses based on this trained model. So here, what you see is the response of one particle neuron in the inferotemporal cortex, predicted by the deep neural network, which is shown in red, and the actually measured response intensities of that neuron to these images. So on the horizontal axis, we have these 1,600 unseen test images, and on the vertical axis, we see the intensity of the response. And although these images are test images, the match between the two, between the artificial neural network and the biological neural network is, I would say, it's quite striking and impressive. And if we do another exercise on how well the different layers of this deep neural network correspond to different layers of the visual stream of the monkey brain, what we see is that when we try to predict V4 responses, which is an earlier stage of processing than IT, or when we try to predict IT responses, then what we observe is that lower layers of the deep neural network can predict these V4 activations better than for IT activations, inferotemporal cortex activations. So it sort of delivers this comforting feeling that the sort of hierarchical processing that is there in this deep neural network somehow corresponds to the hierarchical processing that is taking place in the brain. So then the second question comes. Since responses of artificial neurons fit the responses of biological neurons, biological neural responses so well, can we drive these neurons to peculiar little corners of the activity space solely by somehow engineering the stimulus? So what we do here is that we use, I mean, not we, this is coming from Jim DiCarlo's lab. So they are using deep neural networks to identify those stimuli that selectively drive individual neurons in the V4 of the monkey visual cortex, and then we design these stimuli so that we, based on our artificial neural networks, we expect sort of, let's say, the strongest activation possible for that particular neuron when we consider sort of all the possible stimuli that is constrained to the receptive field of a neuron. So the receptive field concept is shown here. It is the sort of area in the visual field where a particular neuron, to which a particular neuron is sensitive to. So these circles represent individual neurons' sensitivities, and from different animals, we have a bunch of neurons, each with a different, little, slightly different receptive field structure. So here would be one goal that one might want to achieve. This is the sort of the maximal neural drive paradigm, where one is trying to find a stimulus that is such that when we consider neuron 1, then the stimulus is such that the stimulus drives this neuron as much as possible, irrespective of how the other neurons behave. Okay. Another task would be the so-called one-hot population control paradigm, where your goal is not simply drive the neuron 1 as strongly as possible, but with the constraint that all other neurons remain as silent as possible. So these are two possible sort of optimization criteria for image patches that one can do in a deep neural network. So because the deep neural network is a differentiable system, one can find the specific image that would achieve this maximum neural drive goal or the one-hot population control goal. And once we do this, then we can check whether this design stimulus that is only engineered based on our artificial neural network will elicit similar behavior from our biological neurons. Okay, so here's in a nutshell the result of this maximum neural drive paradigm. So when you provide the neuron with natural images or some artificial images which were actually originally thought to be the optimal drive to these V4 neurons, then what we see is that we can. So there is a certain amount of drive that is provided to this particular neuron that we record, and we can actually predict the firing rate of the neuron based on our deep generative model. So the predicted and measured neural firing rates lie on this identity line pretty nicely. The spread of activations is pretty wide. So the blue points and the purple points, you see that it can go from minimal activation to pretty strong activation, and then we can check those images which were specifically designed to drive our artificial neural network to a high drive. And what we see here is that it is indeed true that although sort of the image contrast, which is a primary determinant how strongly a particular neuron responds, the fact that we engineered these stimuli is actually driving to a spot in the activity space of this neuron that is actually not achievable with this large set of natural images. So this is a proof of principle that that what we obtained there in this artificial neural network model is actually predictive with respect to completely new stimuli that are designed to be rather extreme stimuli for our neurons, for our visual system. If we consider the one-hot population control, then again we can do a similar exercise. And now it's more instructive to show not only one neuron but 8 different neurons, so that we can see that sort of the activation of a particular neuron is really sparse in the sense that the stimulus that we optimize with this one-hot population control cost function is really selective for that particular neuron. So for every single neuron, we can find this one-hot population control stimulus. And what you can see here is that once we design this stimulus, it can actually nicely achieve some sort of sparse response pattern in this set of 8 neurons. Obviously, this is a much tougher task, being really selective to that particular neuron. But I would say that this is still a pretty impressive result. Now let's go to a by now well-known feature or characteristics of deep generative models, which is the adversarial attack idea, whereby we test an artificial neural network with different images, like this one. The question is what is on this picture, and the answer, if classified correctly, then this is a school bus. Now, the adversarial samples are tiny, unperceivable manipulations in the image, which actually are such that can result in sort of perceptually identical images, but such that when we ask the very same question, the answer of the deep neural network will be ostrich. And this works pretty well for other images, like you have a pyramid, then you add a different pattern of perturbation, and again you will get an ostrich, and you can do it infinitely many times. So this top of the line deep learning architecture, which is actually matching human performance in simple classification, can be attacked by these adversarial samples quite reliably. So the question is this: if we were so good at predicting neural responses with these deep discriminative models, so the nervous system will follow the same sort of mishap of the deep discriminative models, or whether the nervous system will not fall for this adversarial attack. So again, Jim and his colleagues did this beautiful experiment where they identified a bunch of images, the so-called challenge images and control images. These were identified based on this. When if you could predict the neural responses based on with our AlexNet, then these are the blue points. Then these are called the control images. While for those images, those set of images where AlexNet failed to predict reliably the neural responses, will be called challenge images. So it's a behavioral, it's a characterization based on the behavior of the artificial neural network itself. So then what we can do is this: we can measure the accuracy of the monkey, and then we can record the activities of neurons in this during this stimulus presentation. And then so here on the different horizontal blue lines are the activations of individual neurons in the inferotemporal cortex. And so here you see altogether 7 neuron activations. And these little gray boxes correspond to time windows where we try to do decoding of the image based on the neural recording. So for different time points, for different time windows, we can decode the content of the image with different level of accuracy. And at the time when the decoding performance reaches the level of behavioral performance shown by the animal, this is the time that we call the object resolution time. So it characterizes the neural responses such that when the neural responses contain sort of as much information that we can decode with as high reliability the image content as the monkey does, then this will be a specific signature of the neural code. Now, if you compare this object solution time for the control images and challenge images, what we find is that the object solution time will be markedly faster for the object solution time will be markedly slower for challenge images than for control images. So it shows us that somehow the nervous system indeed identifies these challenge images as really more challenging. It takes more time to achieve a neural code which contains as much information as the response for control images. So finally, we end up here is that we demonstrated in a couple of experiments that this computational architecture of deep discriminative models is pretty capable of doing quite fancy stuff, and one can still ask how specific this architecture is to this high level of efficiency that we observe when we try to predict neural responses. So whether the computational principles and components that we list here are really necessary for achieving the sort of performance that we see in the nervous system. So just to highlight that at least one aspect of this setting is not a necessary component of the artificial neural network to be as powerful in predicting neural performances is a recent paper by Dan Yamins and Jean DiCarlo, where they used an unsupervised method to try to predict neural responses. And just to cut this story short, what they show is that when they try to predict the performance of IP neurons with this local aggregation unsupervised method, then they can actually match the predicted performance of this local aggregation to the supervised setting. And so I will then summarize here this part on deep discriminative models by just pointing out these sort of messages, and I will stop here so that you can ask questions.
Okay, thank you so much for this first part. So just to mention, the participants have had the tutorial at the School on Computer Vision, where they've seen the adversarial example. So it's a good match with your talk. Thank you. So I will just put the questions up. And okay, so I hope you can see them. Okay, so first question, do you think integrating both AI and neuroscience into a field of general intelligence will boost the progress of our efforts for understanding intelligence? What are your views on this?
Yeah, that's a beautiful question. So I think that there's a really fruitful collaboration or cross-fertilization between machine learning and neuroscience, and I think that this cross-fertilization goes beyond the basic claims like attention, which is a neural mechanism observed in complex tasks, which affects neural responses, seem to be important for complex tasks in AI. I think that further insights into what actually attention is in neuroscience will have a positive contribution in AI as well, and this positive contribution will actually bring us closer to more advanced applications. So, for instance, in my view, based on the research we do with mice and monkeys, attention is sort of not a vanilla mechanism to put a spotlight of attention, or a spotlight on subspaces of the visual field, so that we can just focus on the subfield of the visual field. Rather, the attention is a more sophisticated mechanism, which is directly related to inferring task variables than to perform a task.
That's very interesting. What do you think about the role of backpropagation or hierarchical finite difference learning in the brain?
Oh, yeah, this is an amazingly hot topic in neuroscience these days, and yeah, I think that this is again one point which is super inspiring for neuroscience. So historically, backpropagation was considered to be a no-go area for neuroscience, because backpropagation assumptions are hard to consolidate with the mechanisms that are available in neuroscience. But because of the fact that these computer vision tools and other machine learning tools are so amazingly effective in performing tasks like classification, and also amazingly successful in predicting neural behavior. So this way, quite a bit of effort has been devoted recently to sort of lift this barrier that backpropagation is a no-go thing. So more recent studies suggest that dendritic computation, so the neuron which is pretty much an elaborated pipe hose with a cell body and a tangle of little branches, and these branches are called the dendrites. And recent work has highlighted that the computations that are there in the dendrites can actually make backpropagation feasible in neural networks, biological neural networks as well. So I think that there is certainly a perspective for backpropagation, and we learn a lot about these efforts.
Great, thank you. Many machine learning papers speculate on connections between deep models and the biological brains, very often without significant arguments from the neuroscience side. What is your view on this?
So I think that so as you will see in the second part, I argue that the sort of architecture that I presented here, these deep classification networks, are not really the ultimate tools for understanding human or primate vision. But I think that this research program that Jim DiCarlo and a bunch of other people like Diko Kyrgyzkert did is absolutely instrumental, that they actually progressed as much as they could with these architectures, and we see the limits of these architectures. We know how much capable they are of, and then as this last study highlighted, you also see their sort of limits on accounting for these adversarial images. So I think that the progress as machine learning progresses in neuroscience as well, state of the art in computer vision, where the deep classification networks, but more recent efforts are highlighting that other unsupervised tools are also capable of addressing complex image statistics like natural image statistics.
So maybe just one more, and then we continue with the second part. Do you think the role of learning is central in the brain, or is it inner structure and models that are dominant?
I certainly think that learning is key there. There are some ecological arguments for this that pretty much as much as one could encode in the genetics can be done so, but certainly the sort of complexity of stimuli that we are faced with, whether it's language or visual stimuli or some other domains, it is not really a good idea to try to encode it genetically. So learning will have a key contribution to that.
Okay, great. So let's continue with the second part. Please do present again. Maybe you can, because I think we are not in the right set layout yet.
Okay. How is it now?
Let me see. Could you please resume in Google Meet? Yeah, if you try again, that should work.
Okay.
Yes, that's fine. Perfect.
Okay.
Thank you.
Okay, so when we go to another computational framework, again we need to list the sort of components that this computational framework relies on. When we speak about generative models, then the computational goal can be phrased as learning the data statistics. So here what we do is this, just a very coarse-grained math to support this idea. When we learn a generative model, then the goal of the learning is to be able to estimate the stimulus statistics P of X. In the generative model, we try to discover latent factors Y that can actually account for the structure at the level of X. So X is a multidimensional space. Like a million pixels in images, and the statistical structure that is present in X is captured through the presence or absence or combination of latent factors Y. This way, we implicitly define a generative model of the data, since P of X given Y describes what is the probability of the observation occurring when we have a set of latent activations Y. So this way, the factors Y can be considered as the features underlying our observations. So normally, you can describe this function, the likelihood function, as a simple function and some noise model, and obviously you can go with a simplification. Your first attempt would be to speak of linear generative models where the mapping between Y and and x is linear, or nonlinear generative models like variational autoencoders or generative adversarial networks. We will go for the linear generative models in a bit, but before that, we will let me try to motivate the hierarchical structure as well. So right now, the top-down interactions in the architecture as well. So this will be pretty much the only biological motivation that I provide here. I think this is a pretty strong motivation, but I will go for more computational ones later. So when we look at how the visual cortex is built up, you see here V1, V2, V3, and inferotemporal cortex lined up here, which constitute the so-called ventral stream of the visual cortex from V1, V2, V4 up to IT. And you see that the connections between these two are between individual areas is strongly reciprocal. One can make actually claims that in some layers of the V1, the feedback connections, top-down connections, are equal as strong as the connections that are bottom up. The third component of this deep generative model to sort of supporting feature, which is that these deep generative models, since they are not optimized for one specific task, they can learn task general representations. So to sort of motivate this task general idea is it can be done easily through the idea of intuitive physics. So when you observe images like these stacks of little blocks, then so a classification task would be what is the color of the constituent blocks, red or yellow. But once we learn about this sort of features of the environment here, the dynamics of the environment, along with characteristic features like whether red objects tend to be more dense or something like that, then we can actually we can actually make inferences which are much richer than the inferences implied in a single task. So we learn about our environment, and all these questions can be asked. So I want to motivate with this the fact that you want to have a tool which learns about the environment in a fashion that it can make rich inferences. The last one is the nature of the representation learned by this model. So deep generative models are capable of learning probabilistic representations, which I think are crucial for all the delicate tasks that we are faced with. So, for instance, when we are trying to cross a river, we need to make a jump, and we estimate this river being 4 meters wide. Then, because of the fact that we are uncertain about our stamina or physical capabilities, there is some sort of uncertainty whether we would be able to jump across the river. But since we want to survive this experiment, we want to somehow be in a situation where, well, the magical p smaller than 0.05 is achieved. This seems to be a simple task, but let's consider this other one where, based on perceptual information, we also need to actually estimate the width of the river. We don't have access to the ground truth of the width, and when we realize that this is again a probability distribution that we make inferences about the width of the river, then what we are faced with is a situation where we actually need to be able to compute with probability distributions so that we can achieve our goal to be under 5% p, right? So this is just an instructive example, but one can come up with other example from the domain of vision, like in this grid example. So again, we need to estimate the colors of different pixels, like on the right eye or the left eye of that cartoon. And obviously, we are not really prepared. Our brain is not truly prepared for making inferences about the color of individual pixels. So what it does is that we are uncertain about the actual color. We are uncertain about the actual intensity of the pixel. So we use the contextual information to make inference about the color. So what we see there is that the left eye of the girl, we perceive something like bluish turquoise or something like that. Although in fact both eyes are just gray, right? So I just want to argue that when we make inferences about as simple things as the color of an eye, we need to rely on information which goes beyond to the actual pixel activation. And so the uncertainty that we have there at this particular inference will be such that contextual information will affect our final inferences. Okay, so let's go for what we can learn from linear generative models. I will just do this very quickly. So when we look at linear generative models, then we make another assumption about like the structure of P of I, which we call the prior. And if we do that, I will not explain why we do that at this point, but the whole point that I want to make is that with this single assumption that this P of I, the prior distribution, is sparse over latent activations, and then we take we link the mean response intensity of neurons to filter activations in our models, filter activations being the vector y. Then what we obtain is that we can explain why the primary visual cortical neurons have receptive fields which are such that they are oriented, they are selective to specific spatial frequencies, and they are selective to special phases of the stimulus. So these are approximately Gabor filters or edge filters, and I think that it is rather powerful that with these very simple computational principles, one can explain why this happens. If we sort of augment slightly this linear generative model by including the fact that we have an independent variable in visual scenes, in natural images, which is the contrast of the images, which varies independently from the content of the images, which are the features y, then we can explain another host of neural response patterns, like contrast invariant tuning or nonlinear filter properties, such as when a stimulus is presented outside the receptive field of the neuron, which we think that normally the neurons should not even be sensitive to because it's outside the receptive field. What we can see is that depending on the strength, the level of contrast of this surround stimulus, the activation of the neuron that has its receptive field in this this middle disk will be suppressed by the surround stimulus. Again, so without giving really insights why this works such, I just want to say here that this very simple notion that we learn a linear generative model of the natural scenes can explain quite a few things about the behavior of neurons. Let's go beyond. Let's get another insight about the sort of generative model that needs to be represented in the brain. So whenever we spoke about neural activations, we were actually speaking about the mean activations of neurons, which is a point estimate of the latent feature activations, right? So this pretty much corresponds to the best latent feature activations that can explain what we observe in a stimulus. However, when we are presented with a stimulus, then we actually need to make inference, meaning that we need to infer the values of latent features based on our observation X. And this requires us to use the Bayes rule, meaning that we need to sort of reverse the relationship between X and Y that is encoded in the likelihood. And we can do that by combining the likelihood with the prior distribution, which is the P of Y. So what difference does this make? So one consequence of this is that if we want to make probabilistic inference, then we need to actually be able to represent the posterior distribution. So if we want to perform optimally in this probabilistic setting, then our system needs to be such that posterior can be calculated. And another important point is that since this is not a point estimate, so the result of inference is not the best possible feature activations that you can come up with when you want to explain the stimulus. Rather, it's a distribution, a probability distribution. It actually characterizes the uncertainty that is associated with our inferences. So when we observe a stimulus and we want to be able to tell whether a particular edge is present or not, an edge with 45 degrees slant or a horizontal edge, then because our inference is a fully Bayesian inference, then we will be able to tell how certain we are in our estimate. Okay, so then this is cool. Then we might want to ask a number of questions. Maybe you want to ask other questions, but here are the questions which I think are quite relevant at this point. So are there behavior signatures of neural uncertainty representation if uncertainty is represented? Are there structured priors that correspond to the complex stimulus statistics that we learn from natural images? How can neurons actually represent the probability distribution? And what are the signatures of stimulus-dependent posteriors? And finally, how, if there is a hierarchical generative model, how can we distinguish a hierarchical generative model from a discriminative one? So first, I will focus on behavioral signatures. So this is a recent paper by Andreas Solias's lab, where what they did was that they presented the animal with stimuli, and then the animals needed to make some choice based on these stimuli. The task itself is irrelevant at this point. The main point is that the animals need to make some binary decisions. Now, if you record the activities of neurons, then you might want to be able to learn some sort of a likelihood representation that is associated with the choices that the animals make. So since there is a mapping between the responses and the choice of the animal, one can actually use some sort of a deep learning tool to infer the likelihood function that is compatible with making specific choices when the responses of the neurons are something for a particular stimulus. Okay, so we want to learn some sort of a likelihood function that makes this mapping. So this would be a deep learning architecture, and just a proof of principle that it makes sense when you have a high contrast stimulus or a low contrast stimulus, and you make inferences for different neurons for different orientations of the stimulus. Then when we make this inference for the likelihood function, what we see here is that for high contrast stimuli, the likelihood function is much sharper, and for low contrast stimuli the likelihood function is blurred. So there is some sort of an uncertainty associated with this representation that reflects the fact that with low contrast stimulus we are less certain about the orientation of a particular stimulus. And this is all cool, but so this is just a fitting exercise. The question is whether we can actually demonstrate that this uncertainty representation makes sense. And the idea is this: so you can either learn a full likelihood model where individual neurons have the ability to learn different forms of likelihood, or alternatively a fixed uncertainty model a fixed likelihood model whereby for any particular orientation the likelihood is expected to be identical. And with this, one can do the following exercise. One can, for every particular trial, one has a particular likelihood function, and if this likelihood function is such that the width of the likelihood function actually makes sense, then if we shuffle the data such that this information that is encoded in the width of the likelihood is lost, only the peak of the likelihood is retained, then what one can see is that we are worse when we try to predict the behavior of the animals when we do this shuffling. So this experiment tells us is that the width of the likelihood has actual behavioral relevance. Okay, so if we go for the second question, whether the structure, whether we do represent structured priors for learned stimulus tasks, we can go with this experiment. So imagine this: we have three images that are marked with red, blue, and green frames. When we have neurons, for instance, neuron 1 and neuron 2, and we represent their joint activity in this two-dimensional space. for every particular stimulus, we have a distribution of responses for this pair of neurons. For image 1, the red one, the image 2, the blue one, and image 3 for the green one. And sorry for the index mishap. And in this model, the prior would be something that is not conditioned at all on the stimulus, which we represent here with this gray ellipsoid. When we decrease the contrast of the stimulus, which is the second set of images, then what we expect is that our inferences will rely more on the prior, and this way all our posterior distributions, the blue, green, and red ellipses, will converge towards the prior. And in a extreme, when the contrast is down at 1% or so, then our inferences will rely solely on the prior, and this way we expect that the distribution of responses of the neurons will match the prior distribution. So this is an insight when we have situations when we do not present stimulus or just a black screen or a gray screen, then the response statistics is expected to match the prior distribution. Okay, this seems to be a nice experiment because you just have a nicely behaving animal, you present it with some cool movie, and all you need to do is switch off the TV set, and then you expect that you will see the P of phi being represented in the neural activities. Okay, so when the animal observes stimuli, then it is when we record a bunch of neurons, then what we see is the posterior distribution of our inferences. And when we present the animal with a blank screen, then our neurons are solely relying on their prior, and this way the activations that we see for a blank screen, those will be the expectations, that is the prior distribution. So the question is whether there is a relationship between the prior and the posterior. So with probability theory, one can easily make a connection between the two. So if you integrate over P of x, then we actually first we get the joint distribution, the product of the posterior and the P of x is the joint distribution, and then marginalizing over x, we get the P of y, right? So it's basic probability theory. And P of x is nothing else but the trained stimulus statistics. So if we average our inferences, P of y given x over the stimulus statistics, then we expect to have a match to the expectations the prior. Obviously, or not very obviously, but an important insight is that this match between our average inferences and our expectations is only true for well-calibrated models. So for those models which have actually learned the stimulus statistics. So an interpretation coming from this is that if the statistics of the stimuli has been learned, expectations should match inferences on average. So what we can do here is that we can collect a distribution of responses, for instance, when the animal is just observing a switch of screen. So this is a non-parametric representation of the activity distribution, P of y. So these 16-digit words correspond to the activations, the binary activations of the 16 neurons that we record. Okay, so individual channels are shown at different positions of this vector. So the second column in this histogram corresponds to the 16th neuron being active, while the rest being inactive. Okay, so this way we can collect this distribution, and we can then plot these histograms against each other for the blank stimulus presentation and when animals watch some sort of a natural movie. So what we expect is that if it is true that P of y matches the average inferences, that is the average posterior, then activations should lie across this identity line. So the frequency with which any particular state is visited during a blank stimulus presentation and during movie presentation should be identical. So this is what one finds. So these points correspond to the elements of this histogram, since we have a 16-element binary number. We have 2 on the 16th number of points. And what we see here is that these are beautifully aligned along the identity line. Now, if we just calculate or assess the similarity of these distributions, which we can do with a measure called the Kullback-Leibler divergence, then we can just characterize this difference with this. We characterize the similarity between the blank stimulus presentation and the movie presentation with this Kia divergence. Now, an important insight would be that this is only true for a well-calibrated model. So when the animal is young, which is at 20 days after eye opening, after birth, the animal is still not opening their eyes. Its eyes. So at 29 days after birth, the visual system is still expected to be immature. And this way, we expect that in an immature visual system, this match will be not that strong. And this is indeed what one can find. So the prior only matches the posterior with the trained model, and apparently the prior that we record when no stimulus is presented is indeed matching the response statistics when a natural movie statistics is shown when the visual system is mature. Another one is that it is only true for the trained stimulus statistics, so we can use different stimulus statistics to calculate these matches, and the strong match is only true for natural movies, but not other stimulus statistics. Yeah, for the
unfortunately, we are a bit over time, so if we could finish in 5 minutes, that would be great. Thank you.
Yeah, so I will mostly try to wrap up in 5 minutes. Sorry. Oops. What happened here? A sec. So instead, I will just highlight one more important thing here. It's about hierarchical computations in the visual system. So if we focus on 2 levels of computations, For instance, in V1 and V2, let's denote them with the activations in V1 with Y and the activations in V2 with Z. Then, if we look at what's happening when we present a stimulus to the animal at the level of V1, so this is the distribution P of Y when we present a stimulus. Now, in a hierarchical model, in a hierarchical model, sorry, let's just concentrate on the equation. We can obtain this P of I given the stimulus through marginalizing over the activations of Z, and this means that when we observe Y and we measure activity at the level of V1, then this will actually depend on what's going on at higher levels, like in V2. So, yeah. So, sorry. So when we present a stimulus like a square to the animal, and here is the receptive field of a particular neuron shown by this little butterfly thing, then you can measure the activation of this neuron when the stimulus is shown, meaning that when the stimulus is presented, the activation of this neuron will go high. Now, since the neuron is actually relying also on higher level inferences, what the animal infers at the level of V2, that also means that when we infer the presence of a square based on the context, then we expect that there will be some sort of an activation of this neuron, because its activation will depend on this higher level inference. This is what we see there. The neuron which actually does not have anything in the receptive field. So there is no contour, no contrast at all in the receptive field. It will actually show some activation to this illusory contour. The activation will be delayed, but it's still there. And I think that the delay itself is telling us that what's there is some sort of feedback activation that is coming from higher levels at the level of V2 or even higher areas. Okay, and I think that this way, what I did here is that I went over a number of key points in probabilistic generative models to show that generative models can address a number of key requirements of the nervous system, like task generality, representation of uncertainty, and this way the full response statistics of the neurons can be addressed. Not only the mean, there are aspects of the response statistics which I did not speak about, the response variability, the response correlations, which can also be explained by these generative models. Now the problem is that these generative models are